वहाँ पर
साढ़े 8 बजे का सेशन सभी के लिए रिलेटिवली के बाद
भाग 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 हेलो हेलो आ गया बोलना सर आप बोलना हेलो सही आ रही साफ आ रही क्लीन आ रहा है ब्लैक आ रहा है ब्लैक कुछ रहा है थोड़ा सा अच्छा यूट्यूब पे देख रहे हैं क्या नहीं तो जूम नेट की वजह से ब्लैक आ रहा है
तुम सामने बैठना, इधर मत बैठना, नहीं तो तबीयत कम हो जाएगी।
और हमें एक्स्ट्रा आगे सुनना है।
यही है। कोई नहीं आ रहा है।
Looks like not so good that no one is there to talk.
शुरू करें?
ओके सर।
चलिए मैं चीज में कर लूंगा यार, अपने आप इंट्रोड्यूस करवा दो सर। अच्छा चलो डॉक्टर तो आ गया और करो, बुलाया इनको ले आओ, उसको भी गाड़े
थोड़ी साइंस वो भी जान जाए।
सर बुलाया सर, ब्रेकफास्ट में जस्ट आ रहे हैं।
स्टार्ट कर देते हैं।
सिक्योरिटी गार्ड को नहीं सर।
अच्छा।
बुलाया सर, क्या है? आज मैं मतलब सेकंड सेशन होगा 2 o'clock, साथ में हमने अभी के 10 किलो हो गए। वही कहता हूँ जैसा आ रहे हैं पार्टनर सॉल्व अरे
और पार्टी देने में बोला क्या?
तो मेरे इंटरनेट पे ऐसे लगा दो कोर्स पे ताकि पता लगे कि ये भी सुन रहे हैं अभी जल्द से आ रहे हैं।
ओके सर।
ट्रांसफॉर्मेशन करना और probably my colleagues they know it, student frustrate होके वैसे ही ट्रांसफॉर्मेट दे देता है, या if you don't actually you keep on waiting तो mutant ही appear हो जाते हैं. So hundreds of such mutants they appeared, but finally when we did the analysis after some 10 to 15 years, after having 10-15 students यहाँ पर आ रहा हूँ, उन्होंने पढ़ ही दे रखे थे हमें, वो भी क्या करें, बड़े परेशान थे, स्टूडेंट परेशान होते हैं, मेरा पेपर छपे हुए मेरे रिजल्ट, but this was very difficult. So I took all those clones to US to do the analysis. बड़ा खुश हो के बच्चे तो चले गए PhD करके, अब ग्रेजुएट मैं नहीं हुआ, तो पता लगा सारी भगवान ने भी बोला ऐसे नहीं कर सकता, सारे स्पूरियस निकले, none of them produced any analog. So then we got a new strain of hemicholatoxis where you could visualize actually the production of reformycin. हमने जब सिंगल क्रॉसओवर होना है तो उसमें कोई रिफॉर्माइसिन नहीं बननी है, ऐसे क्लोन सेलेक्ट करने and then go for this thing. So that is what we did, and finally थोड़ा टाइम कम है, I will so finally actually that strategy got single crossovers and then confirmed them and went for double crossovers where 82 were swapped 86, right? चलो एक आ गया तो एक चला गया, दो आ गए, तो ये कंफर्म कर लिया हमने कि 86 जो है 82 से बाय सीक्वेंसिंग हुआ है, हाइब्रिडाइजेशन किया, एंड देन अगेन वी हैव हंड्रेड्स ऑफ सच क्लोन्स, एंड देन वी अगेन डिड द LCMS. करने के बाद 3 क्लोन हमें मिले विच रिफॉर्माइसिन का बी का मॉलिक्यूलर वेट इज 754, but then the analog should have produced one methyl group, means its molecular weight should have been 740, right? So you can see here 3 clones, 3, 34, 36 producing reformycin B as well as the analog, but that happened in the year 2012. And almost some 15 or 18 students worked on it. बहुत अभी गाली में देते हैं कि you have put that's what actually them on a very difficult, and we did actually derivatization, we did NMR to really confirm कि वो methyl group missing है कि नहीं, right? So this is actually said NMR report reflected that yes, methyl chromate is really missing, and we have produced the actual NMR, and in order to this happened in the year 2012, and really we were very happy and celebrated it. But then, by then, when we wanted to communicate the paper, nobody would have accepted the paper unless you show the activity of this particular compound against mycobacteria. So we started looking into and did the bioassay. Fortunately, we found out that this compound is effective, but then it should be more effective than what we are anticipating. So we purified this particular compound. That time it was very difficult actually. आप लोग shake flask में करते हैं ना? I will show you some slides. When you go to the fermenter, things are completely different, which I myself did not know. हम छोटी-छोटी flask में हिलाते रहते हैं, उनको बहुत कुछ नहीं बनता है. Even we could not get actually in milligrams the amount that was needed even to do the analysis. So with great difficulty, then by then हम ये भी करते रहते हैं रिसर्च में, जो लोग जरूरी है उनसे स्टैंड चेक, जो जरूरी नहीं है उनसे लड़ लेते हैं, और जो जरूरी है उनसे दोस्ती लगा लेते हैं. हम एविडेंस से दोस्ती लगाया कि भाई अब टेस्ट कर दो that whether it is effective with resistant strains of mycobacterium or not, because that would have been a great thing actually, had it been better than the reformycin B that normal organisms used to produce. So फिर he actually helped us to outsource it to a company where we got actually he only suggested to be conduct for open source drug discovery say resistant strains. So we got the resistant strains and company tested and found out that yes, the compound that the derivatives are something around. भाई खत्म होने वाला है, बड़ी देर बाद आ रहे हो, और चलो बैठ जाओ. Something around 50 times more effective. लेकिन वो बहुत we also did कई बार जब किस्मत रिसर्च में अच्छी होती है तो अपने आप चीजें चलती है, जब भाग्य काम नहीं, वो रखा एक कंपाउंड ना क्रिस्टलाइज हो गया, नहीं तो क्रिस्टलाइजेशन करना तो ये सारे प्रोटीन वाले इतना दुखदाई है, अंजू है ना, बड़ा ट्यूब में अपने आप हो गया कि यार अब और दुखी नहीं करना है, And then the crystal structure was again found out, yes, really, methyl group is missing from the 28th position. And then we also, my colleague actually in US did some binding assay with RNA polymerase and found out that it has better binding ability as compared. So then this paper we published after how many years? Some 10, 20, 25 साल के बाद 18 बच्चे लगे रहे, गालियां देते रहे, एक पेपर छपा दिया, right? But it was a very difficult one. So that is, but then you see after some 20 years, almost same ऐसे commits जैसे पहले मिले थे. Congratulations, you have again tackled a very difficult. But मुझे पता था जो बच्चे मुझे गालियां दे रहे थे, right? And then अखबार में भी आ गया. फिर अगर अखबार में भी आ गया सब कुछ तो लोग बोलते झूठ है. ये ऐसे करता रुकना अपना. पर युगेंद्र को पता था क्योंकि इन्होंने तभी मैंने इनको ले लिया. तेरे लोग ज्यादा विश्वास करते हैं, मेरी ऐसी नहीं करते. So then actually इनको पता था कि ठीक है, बट ये भी कहते थे कि आगे नहीं ले जा पाओगे आप. यहाँ तक तो पहुंच गए. मैंने कहा नहीं देखूंगा. We got the patent. तो हमने कंसेप्ट दे दिया कि स्वैपिंग डिलीशन से इस ऑर्गेनिज्म का भी एनालॉग बना सकते और दूसरा हमने एनालॉग भी. पर ये बाय द टाइम सब कुछ हुआ मैं रिटायर होने वाला था. मतलब देखो अब विडम्बना इतना काम किया रिटायरमेंट है ना. 15 सितंबर को सहारा खुशी खुशी सब कुछ छोड़ के निकला मैं. लैब बंद उसके बाद, लेकिन क्लोन मैंने घर में डाल दिए थे. इनको नहीं जाने दिया, इनको मैं ले गया. I mean, because इतना काम मन नहीं कर रहा था मैं निकोलाटॉक्सिस को छोड़ दूं, है ना. So then we actually how to take things further. इतना काम करने के बाद, इतने बच्चे सैक्रिफाइस करने के बाद कैसे आगे चलना है. Then we sat with couple of my young students, हमने जो कंपनी शुरू कर दी. वो बड़ी दुखदायी कहानी है. कंपनी लगता है बहुत, लोग पीछे से कहते हैं इसके पास में स्टार्टअप है ना. वो बड़ा दुखदायी है. गवर्नमेंट ऑफ इंडिया इज एक्चुअली हेल्पिंग यू बट इट्स नॉट इजी. तो हमने फिर एक कंपनी शुरू की फिक्स्ड इन प्राइवेट लिमिटेड, जिसमें अपार्ट फ्रॉम एनी अदर थिंग्स वी वांटेड टू टेक इट लीड्स टू लॉजिकल कंक्लूजन. जब तक जिंदा है छोड़ना नहीं है और कोई रख लें ताकि बाकी और काम करते रहें. बाई टू एक्चुअली अदरवाइज रिटायरमेंट के टाइम सुपरवाइजर के सारा सामान होता है वो पता नहीं कहाँ चला जाता है. बैक्टीरिया ने इतना काम किया होता है. सो but 4 of these students, very brilliant students who wanted actually to take up this from the startup, we got a grant. And then हमने पहला ये करना था कि let's demonstrate that we can produce another analog. And we knew actually what could be the second analog जिसमें reduction कम हो और hydroxyl group of RNA polymerase can. So ये कंपनी ने फिर शुरू किया DH10. I will not same strategy used. We actually inactivated this dehydratase domain of module 10, replaced it with DH1 of module 2. उससे क्या होगा? 17 hydroxyl, sort of these difficult work biopsy, and finally you see here it produces 17 diet. When we analyze these clones, it produces 17 hydroxy ripamycin B, which turned out to be more effective than the. But ये सारा फिर अभी, and then when we got the analysis done, our collaborator in the US, the analysis found out this organism what happens actually produces 17 hydroxy ripamycin B, and that automatically spontaneously gets converted to SD or to S. And then there is a tautomycin. So there are nearly 6 analogs that are being produced spontaneously within the organism, but they are more effective. We have done preliminary study and we found out that this particular analog, the second one which we have produced, is even better than the first one that we have produced. So then everybody now, in order to take it to a logical conclusion, we are asked actually कि इसमें analog कितना बनता है, ripamycin B कितना बनता है. So all sort of things, it's around 30% is the analog which is being. We did LCMS, we did assays and found out. So this commercialization के लिए ले जाने के लिए आपको प्रोडक्शन कंपनी ये बोलती है, पेपर में तो आप लिख दो जो लिखना है, कोई नहीं चल जाता है. But कंपनी को जब वो they don't believe in papers, जैसे हमें प्रोडक्शन इतनी है, so that they also work out economics. So then these strains we took to the fermenter with the help of intact chemical. We grew them from the startup, did all sort of things and then took it to the fermenter. अब हमारे के fermenter में वो analysis वगैरह तो हम करते रहते, purification सब कुछ. But if you see actually what happened in the fermenter, ये देखो कितना revolution बन रहा है. शेक फ्लास्क में जो आप लैब में करते हो तो आप सभी के लिए जो काम कर रहे हो इमीडिएटली अगर कुछ करना है तो फर्मेंटर में नहीं तो आपको अमाउंट एनालिसिस तक नहीं मिलता है. सो दैट इज व्हाट यू सी. ये जो सारा इतना थिक बना है फ्रॉम 20 मिलीग्राम the production went up to 700 milligram in 15 liter fermenter of both these analogs 24 decimeter rifamycin B and 17 hydroxy. ये देखो पीली हो गई है फ्लास्क. इतना खुश हुआ है कि इतनी rifamycin बन रही है एक टाइम. So all this analysis is being done. We also did actually the kinetics to see what time actually the organism starts producing rifamycin B. Then protocol also was very cumbersome which we were following, but then we came to know that you can follow a simple protocol in order to do. But in order to go far now, we have through this company we have big grant. Now we are getting CIGRI grant in order to take it to upscale the production further, larger fermenter. And then after upscaling, we need to again do Morse models. We need to do the testing. And after having done, then clearly करता है. तो इतना आसान नहीं है. पता नहीं जिंदगी में कई बार ऐसे ही लगता है कहीं इधर दर्द हो गई कहीं इधर दर्द हो गई तब निकलता है. But yes, finally these are the things which we need to do in the next round. Actually, the CIGRI grant which we are getting. But we are very happy actually that clones सारे ठीक है, सब कुछ वही कर रहे हैं जो डिपार्टमेंट में कर रहा था. नाराज नहीं हुए जब इनको हम घर के ऊपर लाए बना दिए और वहाँ शुरू कर दिए तो जैसे कि ठीक है तुम लगे रहो इनके साथ. So that's what actually is the situation and work of nearly. And बहुत सारे लोग मैंने बताया ना कितने स्टूडेंट दुखी हुए. You see actually हम योगेंद्र helped us. Then now Katani from Minnesota, he's very happy and analyzing. He's a chemist. तो आपको भी अगर कुछ करना हो ना, बहुत सारे आप लोग ये सोचते हो अपने लिए बैठ के कर लेना. जब तक आप केमिस्ट के साथ बायोलॉजी वाला या और किसी के साथ एसोसिएट नहीं होंगे, दूसरा बंदा कंफर्म नहीं करेगा, आपका पेपर बहुत अच्छा नहीं बनेगा. So we have a very good chemist who really works with Reproduction and he is very happy. But then ये देखो सारे स्टूडेंट जब फोटो लिखती बहुत सारे तो नाराज हैं, जो खुश हैं उस टाइम एनालॉग बन जाए. Thank you. Yeah, no, the derivative is here. They are all secreted off. The rifamycin B, 17 hydroxy B, and 24. They are secreted off. Then spontaneously they get. Otherwise, we have to convert them by chemically, but some of them they also get converted. So spontaneous, sarah medium matter. That is the one. Yes, please.
So if I've seen resistance is a major problem with the patients who get infected with the drug-resistant bacteria. So for those patients, if this analog is active, then it will be
I'm serious. कुछ तो अपने आप ही करना है. He has gone actually. ये बिगर और वो हमारे स्टार्टअप देखते थे घर के ऊपर है. पर बहुत कुछ बात बता रहे हैं. चलो कुछ के नहीं करना. Yes. फोटो. क्रेडिट देना नहीं है. नहीं जो तो तुम नहीं आओ. टैग भी लेके आके चेक कर लो. ये क्वेश्चन से आप सीरियस नहीं सर. वो आपने माइक्रोबैक्टीरियम प्रोसेस करके आपने टेस्ट किया. उसमें रिजल्ट कितनी टाइम पहले था? नहीं वो जोन में रिविजन से नहीं था. उसमें ये जो इंस्ट्रूमेंट होता है जिसमें you add analog, you add the mycobacterium. If it grows, then carbon dioxide is produced, and carbon dioxide is sensed by the instrument. So that was the उसको क्या मैं भूल गया क्या बोल गया. I showed that slide, but did not explain it. It was not, दोनों के एडिशन में थोड़ा सा क्वांटिटेटिव नहीं हो पाता है, कई बार बच्चे ने ज़्यादा डाल दिया इनार्शन निकाल दिया, but then that was the test which Prema Biotech from Gurugram did. पर अब ये सारा हमने जाना है, दूसरे एनालॉग का भी करना है, so we are planning now to do it in a bigger scale in mass model, probably next.
So sir, naturally also these analogs are being made by the.
नहीं, हम शॉर्टिंग जीन क्लस्टर के साथ खेल रहे हैं। एक्चुअली तो भी बनता है, लेकिन हमने उस क्लस्टर को कम करके ज्यादा करके हाइड्रोक्सिल ग्रुप ऐड हो जाए या एक मिथाइल कम बन जाए, तुम एसिडेट डाल दो प्रोपेनेट की जगह, तो वो जब शॉर्टिंग करनी है तो बड़ी एक्यूरेट होनी चाहिए, फेज शिफ्ट नहीं होना चाहिए, अदरवाइज इट विल नॉट बी एक्सप्रेस्ड। दैट्स व्हाई इट टुक लॉट ऑफ टाइम। अभी scale up going up 15 लीटर में से 1.5 में, now we are next grant में 100 में जाना है, and then we are likely to get in this time another grant where we are not associating with impact because they have 500 liter का requirement, so we need to go to इतना आसान नहीं है ना, फर्मेंटेशन के बाद वो धूप दाब करके उसको अंदर रखने जाने नहीं देते, थोड़ा रात को जाना पड़ता है, दोस्ती बनाना पड़ती है। The research is not only ये जो आप सोचते हो लैब में जाए, आपको भाई चार्ज लगाने के बिंदु को बहुत बार बुलाता हूँ, मैंने कहा चीज़ आपको करना पड़ती है, अंजू को बुलाया था इसी आएंगे, हाँ
जितने ये आपने बताया लोग बताया, अगर एक में रेजिस्टेंस आ गया तो ये सब
अरे 10 साल तो निकल जाएगी जिंदगी, बच जाएंगे 10 साल में जो पढ़ रहे हैं, बनेगा 10 साल में कोई भी एंटीबायोटिक डालो, बट देन वी हैव सिस्टम, तो मैं दे दूंगा क्लोन और बनाते रहो।
नहीं जैसे दूसरा एनालॉग है,
हाँ,
तो अगर एक में आ गया
दूसरा डलेगा,
नहीं दूसरे में भी आ जाएगा या नहीं
आ जाएगा, 10 साल का टाइम है, एवरी एंटीबायोटिक बैक्टीरिया में ना वो भी हमारे से चला, कुछ भी कर लो उनको भी 10-1 साल लगते हैं, कहते देख लेते हैं।
Thank you, sir. Thanks for such a wonderful talk. Now I request Professor Yogen Singh to facilitate Professor Dutal with a memento.
ये तो रिपीट करता है।
Cellular system by Dr. Muthu Ayuvan from CSIR West. He's going to talk on cellular basic biology and application. Cellular is a macronutrient, and very special synthesis of cellular system happens.
So I will be discussing that. Yeah, very good morning to one and all. Thanks for the chair for the kind introduction. So I am from CSIR NIST Thiruvananthapuram, and I will be talking today about the Cellular Proteins, which is a unique biomolecule from microalga, its basic biology as well as the applications. As I told, this is my institute, and for the student we have a SSAR academic program for the PhD admissions. So those who are qualified the national exam like JRF from different CSIR, DBT, all those are all eligible to apply, and we do have 5 disciplines. Chemistry, this is a chemistry cluster lab, chemistry being primary research. There are other activity as well, those are including biology. So I will be talking 3 things. First of all, the basic biology about cellular proteins. As I told, it is a unique because its synthesis has some special features. So all those we will be discussing. And then the cellular protein as such, we found an organic selenium source that is in the form of cellulose cysteine. or selenomethionine or entire selenoprotein, which are non-enzymatic. So that process is a patented process, and then the toxicity, bioavailability, and the bit of therapeutic applications. It is needless to introduce the microalgae to the learned people, but for the sake of student, it is a photosynthetic organism. Microalgae is a photosynthetic organism which fixes the atmospheric carbon dioxide using the sunlight through the energy efficient process called photosynthesis, and it is having array of products, lipids to many pigmented proteins, all those things. And I will be working on omega-3 fatty acid as well, but that is not the topic of today's talk, but more of selenoprotein. And this microalgae will save the future because the farming land and other things are shrinking. So this will give a helping hand for us to secure a sustainable production of food product as well as the other therapeutic compounds. So this is the selenoproteins. It is selenoprotein. It's called selenoprotein because the selenium is co-translationally incorporated into the protein, which is essential trace element. It's quite often it was believed that it's a toxic chemical. Quite often there was an incident in the southern province of China wherein Caspase disease, the bone deformities developed. People tried out, even the physician tried to figure out what it is, but they couldn't. But later they supplemented the selenium that completely cured. The reason being that those region people who have consumed the food, the plants which is grown in the soil, which lack of the selenium. That is why, because the selenoprotein machinery is lack in the plants. However, it accumulates, bioaccumulation is happening. So those regions which is lack in the selenium, that people suffered with this disease. From then onwards, it gained importance, biological importance, that it is having physiological relevance with many diseases. And then these functions is performed by the inside the organism by the class of protein called selenoproteins. The selenoproteins, the classical Watson-Crick genetic code, we used to see 61 amino acids plus 3 stop codon, but it won't behave that one because this selenocysteine is incorporated later. I will come to that bit later. Okay, the key function of this enzyme is it maintains the redox balance and signaling as a primary antioxidant system. Because the one reaction which keep on happening is a reduction. So that is why it's very important to maintain the redox balance. These enzymes are very essential. Although it have other moonlight functions, but this is one of the key function of these proteins. In addition to that, it have a variety of other functions by family of selenoproteins. Many functions are there: thyroid metabolism, reproductive health, immune system, and the anti-defense system is one of the prime function of these proteins. Yeah, something missing, that no issue. There is a selenoprotein family. Many proteins are there. I am working on selenoprotein T, which belongs to the family of thyroidoxin-like superfamily of proteins. And this is the classic motif of cytosine. Followed by U stands for the selenocysteine, which is incorporated co-translationally. Yes, I told you this special. You can see that the TGA, it's otherwise called stop codon. The student, they will be familiar. For them, it will be more useful as well. The TGA in frame, ATG start codon is there, followed by TGA. Okay, so otherwise the protein translation machinery will be stalled at the time because it senses as there is a stop signal. I must stop here, but here it is not stopping. How it is done? Because at the 3' UTR, you can see here that 3' UTR we mapped all those things. That is the story all about. So there is a vessel structure. This is called the SEC machinery or selenocysteine insertion sequences. SEC-IS, which stands for selenocysteine insertion sequences. It is a machinery. These structures finds the RNA polymerase not to stop there. Also, afterwards, it will be recruited a group of proteins which all put together. That instead of stopping there, it brings there is a specific tRNA for selenocysteine. It brings the selenocysteine and incorporate there. That is how the machinery continues. So we did initially, we were finding difficult by using some other homology models. We designed a variety of primers up and upstream and downstream, everything, nothing worked. Then finally we went, because this microalgae genetic information is unknown. So the whole genome we sequenced and found that all the sequences, that is where we got it. And later on, the right primer could amplify the specific regions. So in addition to the specific the sec insertion sequences, there are other elements like tRNA for selenocysteine, that is specific to selenocysteine T. And then other tRNA synthase and phosphoryl tRNA kinase. These are all the machinery associated. All the things in microalgae experimentally we have found out for the first time, and that is an advantage, and which has individually every functions. Since it's known in bacterial system, only the sequence level we have found out that this machinery lacks in the plant system. That is why it cannot synthesize the selenoprotein of its own, but the microalgae can do that. Yeah, then we mapped it. It has the 3 introns and 3 exons, all the things, and at the 3' nuclear you can see the sequence element, which is 94 base pair length, and then this is what once again I am emphasizing that this is the sequence element for the selenoprotein T, and there is an intergenic region of around 163 base pair followed by sequence element is there, which forms the classical hairpin loop like structure, which senses the RNA polymerase not to stop at the stop codon. Instead, it brings the selenocysteine to incorporate co-translationally. So along with there is a 2 organism. One is a Senitesmus quadricoda, other one is Nanochloropsis oceanica. So there is a minor genetic information is known for Nanochloropsis oceanica. So we used that as a reference strain to identify in our own strain. And then the other model like Chlamydomonas also, we have extended this one. We found that it is fairly conserved among these organisms. So whatever we developed, the model is valid, and this which we published in a relevant journal. And this is that sequence element regions for selenoprotein T. We mapped it and compared with other eukaryotic or the non-eukaryotic instead of selenoprotein T as well as selenoprotein W as well. We have done. This is that specific tRNA with anticodon and acceptor loop, all those things. And this is very well conserved between Senitesmus quadricoda and oceanica. And this is diversity of this one. Yeah. So the functional aspect is that in addition to its classical function of maintaining the redox balance, it also found in ER stress. There is from some fungus, people have shown that it is having a role in ER stress. So we found that we just mimicked that, we induced the ER stress by treating with the DTT, and found that the expression is almost 2.2-fold more expressions are found through quantitative RT-PCR. So it does has, but it has an opportunity. We are exploring in that direction further. And then when we come to the second aspect, I am working for CSAR. CSAR, the basic biology is fine for academic interest, but we need to have some kind of product, industry-oriented research. So they want so for that one, we developed this as an organic selenium source. And we carefully evaluated that using ICP-MS. And here you can see the development. It was not a small task. Over several generations, it's being this strain being developed. You can see that the first is the control, and with less lag, it stops and goes to the normal growth phase of exponential growth, followed by the death phase. And similarly, at lower concentration also, when we increase the concentration of the sodium selenide, there is a prolonged lag period. And people even found out that the student lost interest or something, that of late it's again started growing, because that is a physiologically unusual concentration. This organism found that something toxic to it, but however it maintained it. We know from the other yeast and other strains that it is being selenoprotein being done. So it took prolonged lifetime, and now we developed over several generations. It's a sequential things we developed for over several generations, and then later we found that now we have a strain which can withstand up to 1000 micromolar. This is a bit old late data, but up to 1000 micromolar of organic selenium can withstand without any lag. It can grow from like a normal strain. Otherwise, even 5 millimolar is very unusual for the other organisms. That is an advantage. Then later on, we found that it's growing fine. That does it accumulate the selenium inside or not? We evaluated through ICP-MS. We found that almost 1200 ppm of organic selenium. This is with the 500 micromolars, and we have some improvement over that. In addition to that, it also has the essential omega-3 fatty acid, eicosapentaenoic acid, which is having its own clinical relevance as a cholesterol reduction and all those things. And this particular process, we filed a patent. It's a filed only in India, US, as well as the PCT for European Union. Here again, I am not going further, but we found that this strain is withstanding up to that much sodium selenite. What could be its genetic architecture, whether any changes happened, what made them to withstand that? All those we have made a comparative transcriptome, and those data I am not discussing here. So I will move to the scale up. As Professor mentioned, that whatever we have seen in the class is quite different from the actual scenario. So we need to scale it up. We have the 50-liter reactor, photobioreactor. So for all our studies, further animal study and company, this being validated at the company as well. So we are scaled up in each reactor. Maximum of 50 liter can be cultured. So up to 200 liter. It is when we are doing that in the flask, it could be with a little optimization. We can scale it as well. Those same 1000 micromolar concentration, the organism can grow in relatively large scale. I cannot call it as a bigger reactor, but when it works in 50 liter, I hope it will work in 1000 liter as well. Yeah, okay. So the toxicity and bioavailability, we have to test it out, and we do not have in-house animal house facility. So we, before going for that, the microalgae having one intrinsic problem is that it has the recalcitrant cell wall. Whatever unlike bacteria secrete out, it doesn't secrete out. It stays inside. So this it's a 3-layer cell wall made up of cellulose and pectinase, and in between some other layers which is poorly characterized. So we made some effort to carefully optimize the concentration of these enzymes, pectinase and cellulase, and we found that completely without leaching out, we can logically with the concentration of enzymes, we can manage to remove or break the cell wall. Thereby, it is the ruminant animals or other one consumes it, whatever the bioconstituent will be available to the organisms. So after doing this one, and then we went ahead with the animal study to evaluate this organic selenium. Does it have any toxicity? Because selenium it's like a double spot. It's toxic as well, beyond certain concentration. So we went ahead. One is a normal study, toxicity, bioavailability, growth gain, any mortality, all those things. The other one is challenge. We fed this one for about 8 weeks, and then we challenged with we introduced the classical for this protein antioxidant effect is there. So we given the chromium induced anti-oxidative stress we have given and fed that one. It performs very well there. So you can see the data. It doesn't have any toxicity, as well as it's more bioavailable because as evident from the serum selenium level and other different organ. Everything we analyzed through ICP-MS, and we do found that it is accumulate serum in a physiologically relevant level. And then the basic lipid profile, and the outcome of these studies are that it reduces the cholesterol. It doesn't have any effect on DAG and high density and other very low density lipids, whereas it reduces the low density lipid, which is not good for the health. But other effects we do not found any effect on that. Similarly, the hematology as well. The thing is that it elevates the serum selenium level. I told you through ICP-MS we have found that one, and it elevates the selenium level in other tissues as well. And it is very significant increase in WBC was observed. And this organic selenium reduces the oxidative stress. Here I have not shown the data. There are enzymatic and non-enzymatic like glutathione peroxidase, catalases, and those things all we have evaluated. But since the time is restricted, I am not showing that. And the lipid peroxidation is the hallmark of ROS, reactive oxidation species stress. All those when the lipid stress is the oxidative stress started, there is a scavengers also. activating like catalases, MDA level, all those things we have tested out. It seems to be that the organic selenium enriched microalgal biomass having a good potential to act as a human supplement as well as the food feed ingredient supplements. So the conclusion and the future perspective is that we decoded the cellular protein T and it associates set insertion machinery. And if anybody interested in cellular protein P, the family of proteins are there. If you want to express that one, in normal vector we cannot express it because these machineries are lacking in other things. So these microalgae can be used as a heterologous expression system to express other human cellular protein P and other things. And then the structural validation being done, and we enriched the organic selenium, enriched the microalgal biomass, and then the recalcitrant cell wall also being selectively. It's a kind of package. Organic selenium is there, and the cell wall can be removed. So it is ready for dietary food feed supplements. If any industry people list I found here. So if at all in future collaboration, as a perspective that industry people are there, we can have a valid MOU and we can discuss and see its feasibility for their applications, and also any for therapeutic applications appropriate cell line model, and we do not have enough cell line model. Somebody, the like-minded people are there, we can collaborate to go further in this aspect. So with this, I thank my student Reshma. It is her own, she is a very sincere student and intelligent student. All the work which is done, she is also one of the co-inventor of this patent. And Reshma and all my funding and team IMCD for inviting me to share my some of my talk. And thank you all for patience listening. Happy to take
Anybody has any questions? Any question?
One question. Did you look at the liver function test in the mouse models?
No, ma'am, not yet. I am planning to that liver NASH or some fibrosis model is those models someone is asking, we can do it.
Because selenium related that one.
And also we can test it out. That is one of my interests. And also in the endocrine related questions, some specific things. I made some effort because of non-availability of suitable partner. So ours is a biotechnology division middle. It's a chemistry cluster lab in CSIR. We do not have that much biological activity, widespread activity, and animal model is one of the chief labs. In vitro assays, I don't believe those things. So it doesn't work beyond publications.
Right.
So if we have suitable model, we can collaborate.
Right. Thank you so much.
This biomass is now we can scale it up in a gram scale. How much our amount we want to throw the field trial, we can conduct and arrive at meaningful conclusions.
All the best for your future. Thank you. So, if there are no questions, if there are no questions, thank you so much.
Thank you, sir. Thanks for the wonderful presentation. Now I request Professor Kalpana Pai to present a moment to Dr. Muthu.
Thank you. So I will invite the next speaker, Dr. Sashant Kumar, who will be speaking on newer insights into the anti-cancer model of mode of action of Vitamin somnolence.
Okay, so thank you, ma'am, for introducing me, and a very good morning to all of you. So, I'm Dr. Shashank Kumar from Central University of Punjab. I'm working as assistant professor in the Department of Biochemistry for the last 7 years. So I will be talking you invite two lectures that was related to pharmaceuticals. Now, I'm dealing with nutraceuticals. So I will be talking on a much renounced plant that is known as Vitellinia somnifera. And that plant is being used in different medicinal systems across the world. So here I just put the representation of the particular plant that is Vitellinia somnifera. It belongs to Solanaceae. And we know about the pharmacy of, or you can say the pharmaceutical activity of this particular plant. And we heard about this plant in newspapers and in other, you can say, the different literatures. But the PubMed indexed articles here, I just categorize those, and it shows that near about the 25% of the publications are belongs to, you can say, the cancer. And out of that particular 25% of the literature is related to breast cancer, and that it reflects the importance of this particular plant in the field of breast cancer. So here again, this is a slide which just introduces you that the active, or you can say the active clinical trials are there on this particular plant or in product. So it reflects the importance of this plant in this advanced research. And now these are some butanoids, and these are the active anti-cancer compounds present in this particular plant. And I will be talking about this vithaferin A. This is a pharmacologically active ingredient of this plant, and so many pharmaceutical, you can say, the activity are reported for this particular compound, and the various mechanisms have been deduced at molecular level and biochemical level. And this particular slide shows and indicates the beauty of the natural products. You can see here there are so many, there are so many mechanisms of actions of a single compound, and that is of the, you can say, the natural origin. And here you can see the each and every cancer. There are so many cancers that this particular single compound or natural origin is targeting so many cancers. And this is the beauty of the natural compounds when deal with the natural origin of compounds. Okay, so they are multi-targeted. They used to target so many signaling pathways. They used to target so many biochemical aspects of any organism. And the thing that is involved between that, the principle that lies between this particular beauty, I think that is the plant and the animals. We co-evolved. We co-evolved, and this is the thing how, and this might be the reason behind this particular thing, that a single compound is used to target multi-targets. Now, this is the very basic slide that relates to the microRNA, and this is what these are the small non-nucleotide sequences that are regulatory in nature. And the occurrence of these miRNAs, or the percent of these miRNAs I just mentioned here, they are the regulatory in nature, and the most important thing, they are related to their dysregulation is associated with the occurrence of a particular disease, the severity of a particular disease, and this is the fact that initiate, or you can say that opens up a venue to a new era of, you can say, the therapeutic targets. So again, this is a small table that indicates that yes, miRNAs may act as therapeutics because some of the clinical trials have been done. And in the previous slide, I mentioned that here MIR34 that has been approved or in phase 1 clinical trial. So I will talking about the triple negative breast cancer. So breast cancer, you heard about the breast cancer, but the triple negative breast cancer, why triple negative breast cancer? Because it's a subtype of breast cancer. And it's a very dangerous thing, or you can say cancer, because these are the statistics you can find in the figures, and in newspapers you can find it. Here you can see that in India, the triple negative breast cancer, among other subtypes of the cancer, the prevalence is high. And that the second figure belongs to the occurrence of this particular disease among the various cities of our country. And the most important thing here, you can see the hormone-based breast cancer, that the first type of breast cancer, and HER2 positive, and then third is the triple negative breast cancer. Here you can see the incidence of the triple negative breast cancer, and the thing that made it worsens, it is the absence of the pharmacologically active targets here, and as well as the relapse of the disease, the aggressiveness of the disease, as well as the despite there you have got the very less chemotherapeutic ability in different. So here this is what the available treatment strategies in different. You can say the chemotherapy is there, hormone therapy, radiotherapy, and immunotherapy. These are the various strategies to manage this particular disease. And this is here, I just point out some of the problems in the management of triple negative breast cancer. There's a therapy failure, drug resistance, relapse, and metastasis. So these are the very crucial parameters which are related to the mismanagement of disease, or you can say in other terms, we are not able to fully manage the disease. And one more critical point is that this particular triple negative breast cancer subtype used to occur in very younger women, and it is very alarming. This situation is very alarming. And there I just point out some of the miRNAs that had been studied for the management of the disease. And now here the story starts. So there are so many molecular and biochemical, these studies have been done on this ashwagandha or with Arrhenius omnifera. But I just try to elaborate this particular thing, and that's why my topic of this talk was the newer insight into the mode of action, anti-cancer mode of action of the Arrhenius omnifera. So here I started just to, because if you target a particular cancer, if you deliver a drug in your preclinical model or clinical individuals, so these are the three types of cells generally the drug used to go and interact with. First is the normal cell in that tissue environment. Second, whatever the cancer cell is there, whatever the organ you are targeting, and third is a small population present in that particular tumor that is known as a breast cancer cell. So here I just put three different origin of cell lines and just tested the effect of Vithaferin A at the microRNA level. Now for that we just MCF10S cell lines. This is the normal breast normal cells. The in vitro model was the MCF10 cell lines, and for triple negative breast cancer cells we use the MD-MB231 cells, and for the breast cancer stem cells we used MCF7 cells and just produced the breast cancer stem like cells as per standard protocol. Now these are the three studies we have just sequenced the MCF10 treated with the same concentration, and that was the 2 micromolar. Okay, and we just submitted the data, and which will be publicly available after we just publish the data. And first study was related to the triple negative breast cancer. Study second deals with the breast cancer stem like cells, and study third was the Vithaferin breast cell, that is the breast normal cells. So I will be talking about the study one, that is what the effect of Vithaferin on microRNA expression modulation of Vithaferin in triple negative breast cancer cells. So this was the basic experiment. That is what we studied, the effect of Vithaferin A on two different triple negative breast cancer cells, MD-MB231 and MD-MB453, and we got these IC50s that were related to, that was near about the 2 micromolar, and it is well established in the literature. Across the literature you will find near about 2 micromolar IC50 of this particular drug against various cancer cell lines. And this is what the entire just sequencing data I summarized in a single slide, and here we found what the 413 differentially and significantly express miRNAs, and this is the B figure that shows the log fold change and the significance of the miRNAs across this particular sequencing data. And the C, here I put 20 most upregulated and downregulated miRNAs. So, and this is what whatever the miRNAs we got, the enrichment of target gene prediction was done, and the enrichment of those genes in the different, you can say, the gene ontology terms as well as the cake pathway analysis, what was done. And the beauty of this particular thing, we found that yes, in all these things, all the signaling pathways or the gene ontology terms that are related to the breast cancer or aggressiveness or metastasis, these genes, the target genes, were enriched on those geo terminology as well as cake pathways. So this slide shows you the validation of whatever the miRNAs you got in sequence. So we just picked up the 5 up and 5 down regulated miRNAs and validated using the RT-PCR analysis. And out of 10, we just got the validation of only 7 miRNAs, out of which the first you can say 181C5P. So that was the most upregulated miRNA in that particular picture. So again, we thought that whether this particular miRNA is differentially expressed in the normal cell versus cancer cell. So that's why here we have the MCF10A, that is the normal cell line. The expression of this miRNA was checked in this and compared with the triple negative breast cancer cell line, and we got that the downregulation of this particular miRNA was in triple negative breast cancer. So after that, this is the basic experiment that we just transfected these cells to study the functional aspect of this particular miRNA in the given in vitro model. So we just transfected the triple negative breast cancer cell with the help of a miRNA 1C5P mimic, and we got that yes, the experiment was successful because we were getting the miRNA expression in time dependent manner. In the last graph, you can see here, and from here the study starts. Here we just co-treated the miRNA mimic plus the different concentration of methamphetamine A, and it shows that here you can WA denotes 30 denotes to WA IC30 and IC50. So here you can see just co-treatment of mimic with the phytochemical, that is here the methamphetamine A. The expression of that particular miRNA was very high. Now this is the toxicity, time-dependent toxicity of the miRNA 181C. Here you can see the 24-hour, 48-hour, and 72 hours in two different cell lines. A is MB231 and B is 453. Here you can see that the effect of or the cell viability was decreased in time-dependent manner. Now, the mechanistic aspect of this particular mode of action, I just picked the two cancer hormones. That one was the cell cycle and second was the apoptosis. Okay, so here I just picked two of the cell cycle marker, that is CDK4 and cyclin D1, and these two protein markers are responsible for the transition of G1 to S phase of the cell cycle. So first A or B graph here you can see this shows only the effect of methamphetamine at different concentration, that is at 30 and 50 IC50 concentration, and when you just mix or co-treated with the mimic plus various concentration of methamphetamine, you can see the drastic decrease in these two markers, that is the CDK4 and cyclin D1 in this particular graph. So it shows that yes, WA might be working at miRNA level by decreasing by up expressing the levels of the MIR181C and thereby the decreasing or arresting the cell cycle. Again, this is the same experiment and at the protein level. Here you can see the decrease levels of the CDK4 and cyclin D1, and the decrease extent was more when you just make or the co-treat with the miRNA mimic as well as the different concentration of methamphetamine A. Now this picture shows the apoptosis. Okay, so here this vehicular dye, that is for the host dye, the cells were just stained with this particular dye, and the cells were treated with a different concentration of Arrhenius somnifera alone as well in combination with this miRNA mimic. So here in the last slide you can see the mimic plus IC50 concentration, the extent of the apoptosis was greater. So in this B picture here, this shows the decrease in mitochondrial membrane potential, and the green cell shows the dead cell and the live cells of the green cell. So here you can see the extent of decrease in mitochondrial membrane potential. Now again, this is a very not much popular protocol, and this protocol has been utilized for the last 2-3 years or 5 years, you can say. So it is for the semi-qualitative measurement of the apoptotic population of apoptotic populations. So in this particular thing, what you used to do, the instrument is different. Flow cytometer, you used to do the same dye, PIR and XLV, but this type of experiment you can do it with confocal microscopy. So here again, we are getting both the live cells and dead cells, and these are both in the shade of the different concentration of the co-treatment as well as alone. Now this picture shows these generation of reactive oxygen species in the cells, because reactive oxygen species are known as the inducers of apoptosis as well as the toxicity of the particular agent. So this is again anti-cancer mode of action. So here you can see in WIC30 plus MIR mimic, you can see the loss generation was there. But an interesting thing was that we observed in the last thing, that is what we are getting the WIC50 plus MIR mimic. You can see the disintegration of the morphology of the cells, and it shows that this particular combination is highly toxic, and the cells, they just shed off the integrity, you can say. So it shows the potency of this particular co-treatment. Now we started the effect of this particular combination or alone in for the apoptotic markers. And this first graph shows the effect of the alone treatment or the co-treatment on the PARP expression, and second was the BCL2, and these are what the apoptosis related markers. Now, third one is again, this is the Caspase 3 and Caspase 8 related activities. Again, you can see the very huge difference between the alone treatment as well as the treatment at the various concentration of Vitafrin A as well as in co-treatment with the mimic. Here, M stands for the mimic and MC negative control, and WA30 and 50 are the various concentrations of the Vitafrin A. Now, this is again the yes. Oh. Okay, sir. So this particular slide shows the Caspase activity, Caspase 3 and 7. This is an enzymatic activity done by the luminometer. And this is the effect of WA alone or in combination with on the various, you can say, protein markers of the apoptotic cells. So this is the thing. And this is the conclusion slide, and this is what the thing that have been done for the last 2-3 years. And this shows that yes, if you just treat with the cells, cancer cells, it Vitafrin A, it upregulates the MIR 181C FP, and it affects at least 2 of the hallmarks of this cancer, that is what the cellular proliferation, and it is related to the down expression of CDK4 and CC and D1. And second is the 2 of the intrinsic mechanism, that is what the mitochondrial mediated apoptotic mechanism, and second one is Fas ligand and this particular complex mechanism. Here all are just convergent to the apoptotic signals. So this is, and that was related to the triple negative breast cancer cells, and this is what the another study, that is what with the Vitafrin mediated change of the mRNA expression in breast cancer is derived mammospheres. So that we got published a few months back. Just thank you.
So you said that triple negative breast cancer is very aggressive. Generally occurs in early ages. What could be those possible reasons why this is actually growing more in India?
Okay.
Yes,
definitely.
In comparison to US.
Yes. So that was a.
And what is that total percentage which you are actually observing in India?
So, 2 different questions and the answers are very much different. I am not able to speak on the exact answer of these particular 2 questions, because if you talk about the national registry program, and that is what related to the identification of number of the patients of particular cancer in our country, it is very dangerous, you can say, because in the last month there was a standing committee of the Rajya Sabha. They just said that this particular disease should be a notifiable disease, because it shows that we are lacking of the actual number of cases and actual number of, because we are living in a particular country where so much diversity is there, so much food habit is there, and so these all types are related to the cancer, because cancer is a lifestyle disease. So if you don't know the exact number of the disease, so how can you correlate with the geographical region as well as this type of thing? So for that reason, I am not able to answer exactly.
It is somehow because in my studies also I was collecting samples for many facts.
So, excuse me, can I interrupt, please? You can have discussions during the team.
Okay, thank you.
Thank you so much. Thank you.
So the next speaker will be Dr. Umesh
Thank
Kumar.
you, Dr.
Yeah.
Thank you, Dr. Sasank, for elaborating the role of Vitafrin A on as an anti-cancer. Now I request Professor Singh to felicitate Dr. Sasank.
The next speaker, I invite the next speaker, Dr. Umesh Kumar. from IMS Ghaziabad University, who will be speaking on potential epigenetic modulatory effects of curcumin in breast cancer. He is head of the department microbiology and associate professor at that department. Please.
Thank you.
So very good morning to all respected chairpersons and organizing secretary. I'm very much thankful to Dr. Bharati and Dr. Srivastava ma'am for giving me such opportunity to present over here. Although the limitation in private institution is that laboratory infrastructure, but I still used to work over there with UG and PG students. I started my journey from here only, from ACBR under Dr. B.C. Das, where I worked on epigenetic regulation in breast carcinogenesis. My co-guide was Dr. Bharati and Dr. Suresh Hedau. And I'm really fortunate to present over here where I was a student of PhD. Thank you so much for the opportunity. So currently I'm in IMS Ghaziabad University campus, where I'm having around 450 biosciences students. It is more kind of academic, but there are still, some places where I can motivate those students for the research. So here I'm going to discuss the potential epigenetic modulatory effect of curcumin. Lot has been, there are a lot of lectures I was attending over here. Dr. Bharat Agarwal and all other people are already, discussed about the possible effects of curcumin on cancers. But here I'm going to discuss with those epigenetic alterations which is actually also causing, the epigenetic silencing of those specific tumor suppressor genes, in particularly for triple negative breast cancer and double positive cancers. So it is already being discussed, the epidemiology of the global cancer incident. It is still the second most common cancer of the woman in all over the world, or also in India. So I actually checked the DNA methylation is on specific tumors subtypes, double positive, ER positive, PR positive also. But here I took those MCF7 cell line to see whether any kind of reversal can be done of the DNA hypermethylation by this curcumin. So the mechanism was, I tried that because this is a, one of the mechanisms which can be, reversed. So I tried to look into those MCF7 cancer cell lines. I can go to directly to the methodology part. This is how these genes used to get silenced. And I tried that curcumin to actually reverse that mechanism. So DNA hypermethylation is also an early event in various multi-step carcinogenesis. So this actually silences the tumor suppressor genes, and then unregulated cell proliferation occurs, transcriptional inactivation occurs, and due to that cancer used to be produced. So I took several specific tumor suppressor genes. Here I took GST-pi gene, that is an isoenzyme, which is also in carcinogen detoxifications. So this is the structure of that GSTP1, which actually is being silenced. There is a specific, percentage of, that is 44% cell lines are already hypermethylated, GSTP hypermethylated. So I checked that reversal of this DNA hypermethylation by several natural compounds. Here I used curcumin, which is a polyphenol that can act as a hypomethylating agent. So I choose this. I worked on several others also, but here I'm showing on curcumin. So this is a yellow spice, already discussed, active component of the perennial curcuma longa. And I want to go to the flow chart, which I use. Yes. So I use that MCF7 cell line, which is double positive cancer cell line. Then I did the MTT assay for cytotoxicity. And then we have done that specific concentration, different concentrations treatment of curcumin into these 24 hours, 48 hours, and 72 hours. Then we have isolated the DNA, and then bisulfite modification we have done, MSP we have done, and simultaneously we have isolated protein, and then we have checked the protein expression also of the same treated cell lines. So what we observed, this is a MTT assay, and then the IC50 value is calculated. This is that cell line, double positive cell line, which we have given different types of concentration of curcumin, 1, 3, 10, 20, and 30 micromolar concentrations. So this is a DNA domain fragments after a DNA fragmentation assay. And then we have checked that, methylation specific band after treatment, that it has been observed that, that at 10 micromolar concentration, that methylation is being reversed. So then we have gone through with the, Western blotting to see whether the re-expression is occurring. So at 10 microliter specifically, we can see over here that it is being re-expressed by the treatment of the 10 micromolar concentration after 72 hours. So my conclusion is that, this treatment of this MCF7 cells with curcumin for 72 hours at a specific concentration 10 micromolar causes complete reversal of GSTP1 promoter methylation and leads to re-expression of GSTP1. So this can be acted as a biphasic molecule. The concentration higher than the optimum concentration of this curcumin to the GSTP1 promoter gradually reappear. So this means that this is actually working on in the low concentrations. So because at higher concentration it is becoming toxic, and problem is with the bioavailability and absorption into the cells also with the curcumins. So this is my conclusion. Thank you. And that this work has been published recently in Tumor Biology. And this is my private institution, although I'm developing that laboratory at molecular level. It is on Delhi Meerut Expressway, and this is my team. Thank you so much.
Yes. Thank you, Dr. Manish, for such a crisp and very information.
Yeah.
But very presentation. Thank you, open for questions.
Yes, sir.
You said 10 micromolars is the effective concentration.
Yes.
You look at the other concentration and check where the actually you said higher concentration is toxic.
Yeah.
So does it swamp the receptors and therefore it becomes toxic? What exactly? The reason I'm asking is that several years back we have also worked with flies.
Yes, sir.
And it so happened that 10 micromolars was the concentration that gave the best results with respect to lifespan, refractive output and so on and so forth. And even after did not continue with that work.
Right.
But this 10 micromolars, even with the Alzheimer's disease,
yes,
work, there are also 10 micromolars is the concentration that works. 5 doesn't work, 15 doesn't work.
Exactly.
It doesn't. So I mean, what is it that why this 10 micromolars? Irrespective of the organism, perhaps of the cell.
Right, sir. So problem is, that absorption to the cells, more concentration used to get toxics, and I have observed in morphologically in cell lines also, and even at
You didn't show that.
yeah. This is I tried up to the 50 also. The cells used to get tight.
At 50 cell will die.
Yeah.
So cell morphology is changing with concentration.
Yes, sir.
Right. What is happening?
Apoptosis.
The cells are shrinking and at 10 micromolar, what you are seeing, the cells are still at a stage where it can recoup back. But when you go to 20 and 30, as far as reversal is concerned, most of the cells are actually advanced towards apoptosis.
Right.
So naturally, the reversal might not be possible. So that could be one of the explanations.
Yeah.
Looking at the morphology which you are showing here with the cells, that at 20 becomes highly toxic.
Yes.
Okay, I have a question.
Yes.
Last question.
So the extent of providing compaction will be very different across the gym.
Right.
So it's possible that for your locus of interest, you have seen an effect at 10 cycles. While the other loci would require much higher concentration and a much longer exposure.
Yes, and in case of several, different concentration, it is different. Thank you.
All right, thank you very much.
Thank you, sir. Thank you so much. Thank you, Dr. Umesh, for elaborating the role of curcumin on breast cancer. Yeah. Now I request Professor Kalpana Pai to give a moment to Dr. Umesh.
If you can, because you were also my mentor kind of thing. Just because Dr. Mukesh was your friend is my good actually connection from the staff. So I actually I don't know. Mukesh ji के साथ बहुत सारा टाइम स्पेंड है.
So next talk is Dr. Upadhyay, and she is in the Department of Zoology. She is going to talk on the effect of small molecules on protein folding landscape.
Yes. Good morning, everyone. I'm going to talk about effect of small molecules, certain small molecules that can act as chemical chaperones on protein folding landscapes. So, proteins are the most diverse and complex biological molecules. And inside the cell, proteins are synthesized on ribosomes as linear chains. However, these linear chains can be as long as 1,000 or more amino acids. And therefore, folding of these proteins inside the cell is a fundamental question that still needs to be answered. Not just that, these proteins must be kept in a proper three-dimensional conformation to be biologically active, and the cell expends a lot of energy to make molecular chaperones and other helping proteins that would help certain proteins to fold. And in case these proteins are not able to fold, in which case they would aggregate, and these aggregated proteins can lead to certain diseases, for example, neurodegenerative diseases like Alzheimer's and Parkinson's, where proteins like tau and alpha-synuclein, they aggregate. And apart from that, even in metabolic disease like type 2 diabetes mellitus, it has been seen that there's a protein amylin which gets aggregated and causes beta pancreatic cell death. Therefore, the cell has to expend a lot of energy to keep the protein homeostasis, maintain the protein homeostasis, because this is crucial for the cell to survive. Now, as I talked about these linear polypeptide chains, they have to fold into the proper biologically active three-dimensional conformation. These linear chains undergo folding and they travel through an energy space. This energy space can be shown in the form of a funnel, where the horizontal axis is basically the entropy of the molecule, the flexibility of these linear chains, and the vertical axis is the enthalpy or the energy, which slowly comes down as the protein explores all the conformational spaces and forms native contacts and comes down in energy and forms the most stable native conformation. However, as on the left-hand side panel, you can see the funnels are not very smooth. For most of the proteins, especially the proteins that are longer than 100 amino acids, this folding is by itself, the folding is difficult, and therefore the terrain of this funnel is quite rugged. They have to make their way through this rugged terrain in order to reach the final native conformation. On the right-hand side panel, you can also see that proteins that tend to misfold, they generally aggregate because they are not able to slide down through this funnel to the native conformation, and on the contrary, they would go to some local minima, energy minima, where they would make some non-native contacts, and because of that, they would aggregate either into amorphous aggregates or into amyloid aggregates. So therefore, for the cell, it is imperative to make sure that the second law of thermodynamics, that is the free energy of the folding of the molecule, must be ensured. And this is where two components for this free energy comes into picture. One is the enthalpy of the folding, and the second is the entropy of the folding. All right. So from here, we know that inside the cell, we have a lot of proteins called molecular chaperones, like heat shock proteins, an entire repertoire of these molecules that would help these any newly synthesized protein to fold. Apart from that, we have also found out that there is literature that mentions that along with these molecular chaperones, there is a whole repertoire of chemical chaperones that might also help proteins to fold. In vitro, these have been studied, and in the category of chemical chaperones are osmolytes. These are small organic molecules present in the cytoplasm that basically help the protein maintain its osmolarity. Apart from that, so the category of these molecules are sugars, amino acids, and methylamines like TMAO. So there are studies that show that these molecules stabilize protein and therefore must be helping the proteins to fold. What we asked is that what is the mechanism by which these chemical chaperones help protein fold? Also, we asked whether all the chemical chaperones that have been identified, whether they do this by the same mechanism, or whether different chemical chaperones would have different mechanisms by which they help proteins to fold. So as I mentioned in the protein folding funnel, we have two components for this folding. One is entropy and the other one is enthalpy. So if there is a protein that has a difficulty to fold, either it has got trapped based on its enthalpy, or it has got trapped because of an entropic barrier. And this is what we wanted to ask for different chemical chaperones, taking a few test proteins, that whether the folding barrier, whether it's entropic or enthalpic, which kind of small molecule is going to help that. Now, why I'm talking about this here is because in case we understand how these mechanisms are, then we can probably design de novo molecules that can help in preventing protein aggregation and stop those diseases, or at least, help these proteins to form correct conformation and not go into aggregates, because of which we have these kind of protein misfolding diseases. So the two test proteins that we have used, first and foremost, we have used maltose binding protein. There's a double mutant of that, which is established to be a slow folding protein and has a kinetic barrier from the unfolding to the folding properly folded stage. And to corroborate our results, we also checked the same small molecules on the GFP protein. The candidate chemical chaperones that we have used are trimethylamine oxide, which is a proven chemical chaperone, and along with that, we have used trehalose, proline, and serine. So the first experiment, the first question we asked is, how can we figure out if the double mutant maltose binding protein, what kind of a kinetic barrier it has, and what kind of assistance it is getting from TMAO. So here on the left-hand side, we have this crystal structure of MVP, where it shows the two residues at position 18 and 296 that we mutated into cysteines, so that we can have, and we know in the crystal structure that these two residues are closer to each other in the folded form. So we put cysteines there, and we carried out quantitative cysteine reactivity. What we did basically in this reaction is that we unfolded the protein, reduced it, and then we allowed it to fold in oxidative conditions in the presence or absence of TMAO. After certain time intervals, we quench the reaction using biotin malamide, because of which it will, the biotin malamide will go and bind to the unreacted cysteines and will quench the reaction of disulfide formation at that point. And then through immunoblotting, we were able to see the effect of TMAO. So compared to spontaneous folding, when we looked at the effect of TMAO, we found that the disulfides between 18 and 296 were forming much faster in the presence of TMO as compared to in spontaneous folding, telling us that in the presence, in the environment of TMO, the protein backbone is coming closer, and therefore the disulfide bonds are forming faster. So this gave us preliminary evidence that TMO is helping the protein fold by reducing the entropic barrier, because it is reducing the flexibility of the peptide chain, as a result of which it is leading to a faster conformation attainment of the protein. To deconvolute it further, we carried out Arrhenius analysis, where we were able to differentiate between the enthalpic and the entropic barrier. So in the Arrhenius analysis, we took two different concentrations of TMO and compared that with that of spontaneous folding across 7 different temperatures, and compared that or plotted that against the refolding rates. And by fitting this data into the Arrhenius equation, on the right-hand side panel, you can see the rho value, which I highlighted in yellow probably. There we can see that the rho values are decreasing. So as the rho values are decreasing because of the presence of TMO, it shows that the entropy is being controlled by TMO. And similarly, we have also shown that the disulfide mutant, where the two components 18 and 296 are coming closer by itself and causing decrease in entropy, that is being affected less by TMO. So this shows this gave further evidence that TMO is basically working on the entropic barrier and decreasing that. So based on these evidences, we carried out a high throughput screening to investigate if there are, apart from TMO, there are other molecules, small molecules, that would do the same or similar kind of function for the DMMBP protein. On the left-hand side, we have the heat map for different small molecules. So we have taken multiple amino acids, some sugars, and tried to see the refolding rate as well as the refolding amplitude to see what kind of effect these molecules would have on DMMBP folding. And from this, we selected, based on certain parameters, we selected, apart from TMO, we selected serine, proline, and trehalose for our further studies. On the right-hand side panel, you can see these are the refolding traces of DMMBP in the presence of different amino acids on the top right-hand side corner. And at the bottom, we have the effect of different sugars on DMMBP folding. So from here, from the heat map, from the data that we had got, we selected 2 amino acids, proline and serine, and we selected trehalose as well as TMO that we were originally working on, and carried out the same Arrhenius analysis to see what kind of influence they have and whether we can deconvolute the enthalpic and the entropic barrier between these two, effect between these osmolytes. So on the left-hand side, we have the Arrhenius analysis as had been done previously for TMO. And on the right-hand side, again, we have the highlighted column where we have the rho values, which tells us that trehalose has similar function as we had for TMO, whereas proline and serine do not work in that way. On the other hand, proline and serine the way they are, bringing faster folding is by working on the enthalpic barrier. So we clearly see that different chemical chaperones have different modes of carrying out their action of, helping proteins to fold. Prior to this, there were no evidence which would specifically tell, comment upon what kind of effect a particular protein, a particular amino acid or osmolyte is bringing in order to fold the protein faster. All right, so clearly we could, figure this out, that from the threshold value in these two boxes that you can see, serine and proline are lower because they are showing assistance by changing or reducing the enthalpic barrier, whereas TMO and trehalose, they are helping the protein to fold by reducing the entropic barrier. To corroborate this further, we carried out single molecule FRET experiment to see what kind of populations are generated in the first 100 seconds of refolding of DMMBP. In order to do this, we selected 3 pairs of, we made 3 pairs of cysteine mutants. Those are mentioned on the left-hand side panel on the crystal structure of DMBP. And we tried to see that as the proteins fold, what kind of populations are generated in the first 100 seconds of the protein folding in the presence of different molecular chaperones versus spontaneous. And on the right-hand side panel, we have the FRET efficiency distributions, which tell us that if we look at the parameters for all 3 all three pairs of mutations, we see that the distribution that we have of populations in each of them are different from what we have in spontaneous. At the same time, if we compare them, which I have in the next slide, if we compare the effect of proline and try to superimpose that on TMAO, trehalose, or serine, we find that for TMAO and trehalose, it is different from that of proline, whereas in case of serine, it is similar to what we have for proline. And again, this was like further evidence at the single molecule level that the mode of action of TMAO and trehalose is similar, and whereas the mode of action of serine and proline were different from that of TMAO and trehalose. Now, at this point, we wanted to know that this kind of categorization that we have between these chemical chaperones, does this hold true for any other test protein that has a folding issue and can be fixed by the same mechanism, or whether it varies from protein to protein? And therefore, we used the enhanced green fluorescent protein, and we carried out Arrhenius analysis for all these for this protein using 24 different chemical chaperones. And we found out, to our surprise, that the mode of mechanism now in case of GFP was different from what we had found in case of DMMBP, which more or less told us that depending on what kind of protein we have and what kind of folding intermediate or folding kinetic trap it is into, the effect of the chemical chaperone would be different. So the kind of entropic barrier removal that we could see for trehalose and TMAO was not present in case of GFP, probably because the structure of GFP is different, and the kind of assistance GFP required for a faster folding was the requirement was different as compared to that of DMMBP. So based on this, we could conclude that chemical chaperones, they function in a much diverse way compared to what was thought earlier, and therefore much more studies are required to be able to categorize this. The conclusion we could draw from our study was that the mode of assistance that these chemical chaperones would provide depend on two factors. One, the kind of refolding barrier that or a folding barrier that a protein would have, and also the nature or molecular properties of these chemical chaperones that are being used. So therefore, any chemical chaperone would can assist a protein to fold either by removing the enthalpic barrier or removing the entropic barrier, and that would depend not just on the on the chemical chaperone, but also on the type of folding intermediate or trap that the protein is into. Based on this data, we can project from here that we should be able to design de novo chemical chaperones that should be able to have a synergistic effect together, one component taking care of the entropic barrier, the other component taking care of the enthalpic barrier, and together they might be able to ameliorate the kind of aggregations that we see in different kinds of proteins, be it amyloid proteins, amyloid aggregates, or amorphous aggregates. And future directions would be to try them out in vivo systems. So I would like to acknowledge my postdoctoral mentor, because this was my postdoctoral work, Dr. Kaushik Chakraborty in IGIB, CSIR IGIB, my lab members and funding agencies, DBT, India Alliance, and CSIR. Thank you.
Any question?
Good presentations. These are we working away from native conformation or these proteins?
Yes, of course. So this is the pathway from where the protein starts to fold. So once it is translated. Now, yeah.
What about the way you are analyzing the protein?
Yeah. So what we are doing.
I think that it is away from the native thing.
I would beg to differ, because what we are doing is we purify the protein, then in vitro we are unfolding the protein, and then we are allowing it to fold. So this is in a buffer system, and it is folding now in the presence of certain other molecules that we are adding to the buffer system.
So those are not a native conformation, right?
So the protein is trying to attain the conformation.
Do we have the CD spectrum or to ensure that the protein and better not to touch naturally protein mutation and it makes a serious conformation change.
Protein, I did not do any protein mutation. Protein was added into the environment to see if protein would have an effect on the folding of the protein.
Folding,
These,
refolding.
yeah.
We have the CD spectrum.
We have the CD spectrum for the fully folded protein. We have a CD spectrum for the unfolded protein. No, I do not have the data here. But for
GFP, you are getting the green fluorescence, right? So that's
Yes,
the
sir.
activity.
Yes, sir. So the activity is there because when the protein is completely folded now, it will show the activity. So what we have used for DMMP is intrinsic tryptophan fluorescence. So as the protein folds, the fluorescence goes up. So that has been.
But GFP, for the folding purpose, epifluorescence, GFP is not the right mode. Many things happen with GFP because of the epifluorescence. So,
We can, that's a good point. So we can try
you
with
need to
other
have a
tests.
CD spectrum, unless otherwise the point of folding cannot be clearly understood at the mind.
All right. So I'll look at
Karina, I have this one.
Yes, sir.
Clear level. So if you have multiple cysteine, do you think it is going to force the correct folding, or do you require a chaperone there?
No, sir. Just we have put cysteine there to see if disulfides are forming faster because the environment has
So
TNO.
faster is fine, but suppose there are multiple cysteines, how it is going to help that correct cysteine is that one and this?
So that does happen, and for that we have cysteine protein disulfide isomerases that change.
That's what I'm saying.
Yes.
So you need the chaperone.
Yes, sir. You would need that enzyme to carry out that. But in case of DMMP, the test protein we have used, we have just selectively put cysteines there. So the cysteine disulfide that is forming, it is that particular 8 and 296 that is forming the disulfide bond, not any other.
Yes, last question.
Okay, I'm a little curious, you mentioned phenolose. So does it have anything to do with the fact that phenolose is a non-reducing sugar and it serves as an antifreeze?
There are multiple sugars that have been shown to, have this effect on protein folding. So in many cases, Chaperones has been shown to do this. I cannot specifically comment on the fact that it is a non-reducing sugar and therefore it would be having this effect. But presence of these polyols have been shown to have, better folding effect on multiple proteins. It has been shown for amyloids also.
Just adding on to that, many of the tardigrades and some of the organisms which can withstand the extremes, they have this, as you rightly said,
Marine animals.
antifreeze and all, yes.
Yes, marine animals and animals, they are in deep sea. They have to survive under pressure. They generally have a higher concentration of TMO, betaine, taurine to take care of, the correct conformation of the proteins are being, maintained inside the cell under high pressure and higher salinity.
Yes.
Protectants in piezo.
Thank you, Dr. Malama.
Dr. Ananya. Now I request Professor Yugen Singh to please present a memento to Dr. Ananya.
That means my photograph.
I'm also a CSR man.
So the next speaker and the last speaker in the session is and she's from Department of Zoology. She will be speaking on heterogeneity in the fat body tissue revealed using single cell RNA sequencing. Over to you, please.
Well, I thank everyone in the audience for sticking around. So today I'll be speaking about the role of the fat body tissue in immunity and reproduction, which we are addressing using single cell sequencing, as the title suggests. So if we look at the interactions between hosts and pathogens, it's like an arms race where if a host successfully defends itself against a pathogen, then the pathogen can quickly counter-evolve, resulting in this kind of an arms race, which is a constant tug of war. And I think Professor Ruplal in the morning very brilliantly explained how quickly bacteria can evolve resistance against multiple defense targets. So now, because of this threat of the pathogens that we have, the immune system that we have is multi-layered. So there are different components to the immune response, ranging from physical barriers, which prevents the entry of the pathogens, to specialized proteins and cells, which can target these pathogens and kill these pathogens. Now, this immunity, howsoever important it is, can be affected by several different factors. It could be our own physiology. So how old are we? How active are we? How fit are we? Similarly, our genetics could also affect our immune response, the environment we live in. And given we are in a nutraceutical conference, the kind of nutrition that we consume can also affect our immune response. So for this presentation, I'm specifically focusing on the effect of reproductive status on the immune response. And if we look at the examples in the literature that is there, more often we see that the reproductive physiology often negatively affects immunity. So ranging from insects to birds to mammals, the reproductively active organisms often have poor immune response. I'm an evolutionary biologist. So in our terminology, these negative relationships that we see between the traits, we refer to them as trade-offs, which essentially means that there are often constraints, which could be physiological limitations. It could as well be the availability of the resources, which could be limiting, as a result of which, if investment goes up in one trait, so in this case, let's say reproduction, it can come at the cost of another trait. So in this case, that would be your immunity. Now we see a lot of the examples, and I already showed you the diversity across which we see this, but we do not really understand that why reproduction and immunity are negatively correlated. So this was the question that we tried to address. The organism that we used was Drosophila Melanogaster. So I think, it's a wonderful organism. We can measure all the different traits in the organism, be it immunity or reproduction. In the lab, we also see that reproduction has a negative effect on the immune response in Drosophila. So if mated and virgin Drosophila females are infected with a gram-negative bacterium, Providencia redkeri, so the way infections are done, so a thin needle is dipped in bacterial suspension and the flies are infected with this bacteria. So when mated and virgin females are infected, so on the x-axis is hours post-infection, on the y-axis is survival post-infection. What we see is that mated females have lower immunocompetence, or they die more because of this infection with Providencia compared to the virgins. So we asked that why do mated females have poor immune response? For that, we focused on the fat body tissue in insects. So it's one of the major organs in the insects, and it controls multiple functions. For our interest, it controls both reproduction and immunity. So we wanted to understand that how is the fat body tissue controlling so many functions? Does the fat body tissue consist of multiple subpopulations where each population has a specific function, or each cell perform these functions and as a result of which they are constrained when they have to control multiple functions? So the hypothesis was such that, let's say under the basal conditions, we have a specific set of subpopulations. When the flies are mated, different subpopulations respond differently. Now their gene expression can change, their cell numbers can change, or the new cell populations may arise due to this change in the physiology. On the other hand, upon infection, there are different subsets of cells which are affected. And when there is a dual stimulus in the case of, let's say, mating and infection, these cell populations are then constrained. And as a result of which, we see a negative correlation between reproduction and immunity. So in the experiment, we collected our samples 24 hours post-mating, and the infected samples were collected 6 hours post-infection. So for this, we took 4 Drosophila, 4 treatment groups, basically virgin uninfected, virgin infected, mated uninfected, and mated infected. We dissected their fat body tissues, and we did something which is called as single cell sequencing. So essentially in this case, a single cell is entrapped in a droplet, which also contains all the reagents to prepare a cDNA molecule. So all the mRNA molecules are converted into cDNA within this droplet. A barcode is added on that cell, basically, and that's how then by computational pipelines, we can understand that what gene expression came from which particular cell. So this is what we did. Now coming to the results. So here it's a 2D projection of a multi-scale data, basically. Each dot is a single cell. Its proximity to the other dot or other cell is dictated by the overlap in their gene expression. Greater the overlap, greater the proximity of these two cells would be. So we were able to identify the fat body subpopulations within our data set. When we were doing dissections, the fat body tissue is extremely difficult to kind of take it out essentially. So there were other tissues which were attached with this fat body, and by using the predefined markers, we were able to identify those cell populations and kind of focus just on the fat body cells. So this is the same graph color coded in our treatment groups. And then we looked at the gene expression across those treatments. What we saw was following, that in the virgin samples, so virgin uninfected are here, virgin infected are here, and we saw that both of them have higher ribosome biogenesis. In addition to that, virgin infected females also showed lower ER stress. On the other hand, the mated uninfected females had lower ribosome biogenesis, they had higher metabolism. And mated infected samples showed lower ribosome biogenesis and higher ER stress. So we wanted to understand what does this really mean for the biology of the tissue. So Ananya in the previous talk talked a lot about protein folding. So when ER stress happens, it's the proteins which are not properly folded. And as a result of which, the misfolded proteins begin to accumulate in your endoplasmic reticulum membranes, because of which these membranes dilate or they swell up. So we looked at that using electron microscopy. So the fat body cells from the virgin infected showed really constricted or the natural ER membranes. On the other hand, in the mated infected samples, these ER membranes were highly dilated. We also saw changes in the ribosome biogenesis. So it basically included genes such as which produces different subunits of the ribosomes and rRNA genes, as well as translation initiation factors, suggesting that if ribosome biogenesis is affected, in the downstream, it will affect the protein synthesis. So we next asked if the reduced ribosome biogenesis results in reduced protein synthesis in the mated infected. So to understand that, we did some Western blotting using puromycin. So it's an amino acyl tRNA analog, which gets incorporated into nascent polypeptides. So higher the incorporation of puromycin would indicate higher rate of translation. And that's exactly what we are seeing here. And when we did some quantitation on these, we could show that virgin infected females had significantly higher protein synthesis compared to the mated infected females. So the model that we worked out was the following, that if infected virgin females can immediately synthesize immune-related proteins upon infection, they have better survivorship. On the other hand,
excuse me, can I ask at a wind up of 5 minutes?
Sure. In 5 minutes, right? Yes. So mated infected females had lower ribosome biogenesis and higher ER stress, because of which they're not able to start the protein synthesis, resulting in poor survivorship. So if we change the protein synthesis during reproduction, then can we really go for better survivorship? So skipping these schematics, if people are interested, I can talk about it later. We basically treated flies with cycloheximide during the time of their mating, and we allowed the flies to recover from this cycloheximide effect and then infected them. When we look at their survivorship, we showed that the cycloheximide treated mated infected females survived significantly better compared to the mated infected females. When we look at the protein production, again, cycloheximide mated infected females had significantly higher protein production compared to the mated infected. We also looked at the production of immune peptides by using some MALDI-TOF. So here it's just a spectra of MALDI-TOF of antimicrobial peptides. And we again showed that the number of peptides, immune peptides that are produced by virgin infected and cycloheximide treated flies is significantly better compared to the mated infected females, suggesting that this is in some sense a plastic response. So again, to put everything in perspective, it's possible that by providing amino acids or changing the diet, we can again maybe influence the translational capacity. So to conclude, mated females upon infection suffer from suppressed ribosome biogenesis. Consequently, mated infected females synthesize less proteins when they are infected, and translation regulation at the time of mating can later direct translation machinery towards infection. So essentially, using fat body for multiple processes constrains its translational capacity. And with this, so this is my postdoctoral work, and I would like to thank my mentor, Professor Brian Lazaro, the scientist in the lab, Dr. Ashley, and my undergrad, Nick, which helped me with a lot of gene sorting. And with this, I'll take any questions. Thank you so much.
Just a suggestion, and if you have taken microbiome also into consideration in the virgin and related
so I think definitely, sir. So with these setup, we haven't shown, but I think there is. But there is evidence that there isn't a huge shift between virgins and mated in the terms of microbiome. So I guess in this context, I guess, but definitely yes.
So what is this is not with respect to the way you are looking at it. Otherwise, I'm asking, like, if, for example, there are infected females, what is their like reproductive output or capacity? Do you see certain kinds of?
So it's highly variable from pathogen to pathogen. For this particular pathogen, if we compare. If we match for the window, because these flies would also begin to die. So if we consider their, as long as the infected females are living and we compare that with the uninfected females, the reproductive output do not change, basically. It doesn't change, at least with this case. There are certain bacteria where actually infected females lay more eggs because we call it as terminal investment. So they are trying to reproduce or produce as much progeny as possible before they die, basically. So.
And another thing which I wanted to ask. Another thing which I wanted to ask you is you did single cell analysis, especially the RNA analysis. Did you also look into what are these different populations, the types of cells?
So. Yes, theoretically, yes. So I have looked at all the markers and what those markers would mean. I didn't have a chance to kind of then basically, I mean, the next course of action typically would be to knock down those markers and see how it's affecting those populations. We haven't gone that far, or we haven't really physically looked at those on the tissue. But in terms of markers and the putative function that the cell population might have, that much we have already done, and that's also included in the paper, basically. Thank you.
What is regarding third body? this fat body does resemble human adipose tissues?
There is similarity in terms of fat storage, yes.
Okay. Now, like second question is related to like after infection, like usually like after mating, what is production will be high?
Yes.
But according to your presentation, it is low. What is the reason?
No, so I'll come to that. So that's where it was a lot more confusing for us to begin with. So their ribosome, when I say their ribosome biogenesis is reduced, it's the gene expression which is reduced. But when I look at the protein synthesis, so their protein synthesis in mated uninfected is similar to the virgin uninfected. So we think that this is what the basal state of protein expression is. That is probably where the homeostasis kind of comes into the picture, and that's the basal level. The fact that they have gene expression or they don't already have mRNAs to synthesize new ribosomes is where we think that the mated infected females are not able to make new proteins.
But you told like they lay more eggs, but in some
with some other bacteria, so we don't know how.
Like even for laying more eggs, also they need more protein.
So a lot of protein synthesis and yolk production happens in the ovaries as well, and we don't know whether how the translation is affected in the ovaries. The other being the translation inhibition that we are seeing is also specific to this pathogen as of now. We are like, one of my projects is to kind of test how general this phenomena is. And then I think I'll be able to better answer your question, basically.
Since the protein synthesis machinery was disrupted, that's why.
Yes. Ribosome biogenesis is disrupted, and that's why we think the protein synthesis. Yes. Yes.
Okay. Thank you so much, Dr. Vedika. So I think I interrupted you in there so that we could have enough questions. Thank you so much for your presentation.
Thank you.
So we wrap up this session.
Thank you, Dr. Vanika. Now I request Professor Ruplal to facilitate Dr. Vanika with a memento. Thank you, sir.
So this session ends now, and the announcement.
Now I request Professor Anvi Srivastav to facilitate the chairpersons. Please, ma'am. Professor Mallikarjun, sir, please, Professor Mallikarjun, please come for.
What